multiplication of TMV. The bit what we are going to discuss it today. Tobacco mosaic virus. Friends, as you know, TMV is a plant virus which infects especially tobacco plant. Okay. The infection of TMV virus causes characteristic pattern such as mosaic-like spots or mottling and discoloration on the leaves. Hence the name mosaic disease is given to this. Okay. The TMV was first virus ever to be discovered in the field of plant virology. Okay. So friends, the mosaic pattern what we are talking about. So here the diagram. Okay. The pictures are here. So you will see this one is a normal leaf and this one is the infected one, on which alternate lot of light green and dark green patches are there, isn't it? In light green patches you will find chlorosis. So chlorosis is the symptom developed after infection, in which what happens, disintegration of or degradation of chlorophyll pigment takes place. Leaves are green in color because of presence of chlorophyll pigment, and if these chlorophyll pigments are degraded, then ultimately the leaf turns to yellow, isn't it? So that process is called as chlorosis, isn't it? So this is the tobacco plant in the field. Here the spots what you are getting here, some leaves are of healthy type, and here on this leaf you will find some symptoms. Okay, friends, before we go to its multiplication process, so we should know some certain things of tobacco mosaic virus. I mean of structural aspects. Here yesterday we have discussed these facts. Okay, it's a rod shaped virus. Okay, and having the diameter of 300 and 180 nanometer diameter. So the number may little bit vary. The number of capsomere is approximately 2130. Yesterday's lectures we have the number was 22. Okay, the number may little bit vary from reference to reference, doesn't matter. The molecular weight of TMV particle is 40 into 10 raised to 6 Dalton. Friends, you have to see this 10 raised to 6. Okay, the molecular weight of this particle is 40 into 10 raised to 6 Dalton. Each capsomer means the protein cover present of which the capsid is made up of. So the capsomer is made up of what you can say 158 amino acid. Capsomers arranged in a helix around the central hole of radius 4 nanometer diameter. Okay, so in center one halo is present, or what you can say empty gap is there. So that is called core, and it is of 4 nanometer diameter. So these are the structural aspect. So as far as genome means its nucleic acid. It has a single stranded unsegmented positive sense RNA. Okay, so active RNA you can say here. So positive sense and negative sense हे आपण पुढे पाहणार आहोत. It contains about 6400 nucleotides. Okay, so the genome, that means RNA of TMV, contains about 6400 nucleotides and having molecular weight of about 2.1 into 10 raised to 6 Dalton. Okay, here you have to, this is not 106 but it is 10 raised to 6. Okay, the RNA encodes 3 essential proteins. Okay, the proteins responsible for growth and multiplication of TMV virus. So the RNA encodes three essential proteins. RNA, what are those proteins, friends? RNA dependent RNA polymerase. Okay, RNA dependent, RNA वर अवलंबून नसणार RNA polymerase. RNA polymerase means RNA ला मल्टिप्लाय करणारं जे काही एन्झाइम आहे त्याला म्हणतो आपण RNA polymerase. Okay, the enzyme responsible to multiply or to copies RNA. So that is called as RNA polymerase. It is also called as replicase. Again, movement protein. So TMV virus moves from one cell to other cell. For that purpose, one protein works there. So that is called movement protein and coat proteins. Coat proteins, you know, capsid. Capsid is nothing but it is a protein part. So these are the three essential proteins encoded by this RNA of TMV. Now the transmission takes place by mechanical, biological, and physical means. Okay, so mechanical by through agricultural tools, biologically through seeds, pollen grains, and physically through winds and water. So transmission is transfer of this virus from infected plant to healthy plant. So this is what there are three ways of transmission: mechanical, biological, and physical ways. Okay, so friends, multiplication cycle of TMV. So multiplication is the process of where the virus increases its number. Okay, virusच्या copies वाढतात त्या ठिकाणी, number वाढतो. So this process in case of TMV that we are going to have here is very what you can say, actually it is a complicated process, okay, to understand, but we will try to see it in a simple way. Okay, we will try to study it in simple way. Actually it is very complicated one. The process of multiplication of TMV inside the cell is little bit complicated, but we will see it in a simple manner. So the multiplication of TMV here it involves first step. First, for the process of multiplication, virus has to enter into the host cell. Friends, okay, getting the point? मल्टिप्लाय होण्याच्या अगोदर त्या व्हायरसला सेल मध्ये जावं लागेल, होस्ट सेल मध्ये. सो बिकॉज आउटसाइड द होस्ट व्हायरस कॅन नॉट मल्टिप्लाय, आउटसाइड द सेल व्हायरस कॅन नॉट मल्टिप्लाय. सो फॉर दॅट पर्पज व्हायरस हॅज टू एंटर इनसाइड द होस्ट सेल. देन second step is uncoating. Uncoating means what? The TMV has protein coat, we call it as capsid. Okay, so the capsid needs to be what you can say, it needs to be that coat needs to be removed. Okay, so that process is called uncoating. Again, intracellular development. After uncoating, certain development occurs there. Assembly. Okay, assembly means it is also called maturation. Here, what happens during the multiplication of this TMV? Nucleic acid met RNA multiplies. Okay, and forms its multiple copies. RNA multiplies and forms its multiple copies. Along with that, for the process of assembly, final assembly of virus, a protein coat is necessary. So the protein coat needs to be prepared or synthesized there inside the host cell. So nucleic acid is there and protein is also there. Now both these component needs to assemble for final formation of virus particle. Okay, so that is called assembly, and the last step is release of virus from the host cell. Okay, so these are certain steps in the multiplication of TMV. Okay, next, the first step is what? Entry of virus particle into the host cell. Okay, so entry of virus particle into the host cell, how it takes place? So we know, we have seen there are various ways of transmission. Transmission means entry of virus from diseased plant into the body of healthy plant. So TME cannot directly enter into the host cell. Okay, so it requires damage to the plant cell. Plant cell ला damage, एखादी wound झाली, जखम झाली, तर so through that wound, the TMV enters into the healthy cell. It enters through the gaps, breaches, which are there in the cell wall. Okay, so some gaps remains there. Gaps are there in the leaf cell, especially. Okay, because in the leaf we have different types of cells there. If you see the leaf anatomy, palisade cells are there, spongy cells are there. In spongy cells, here you will find lot of ear gaps, isn't it? Ear spaces are there. So these ear spaces, through these ear spaces here, or what you can say, through these gaps, here virus enters. Okay, next. Uncoating. Now, once virus enters into the cell, then it has to remove the outer covering. Okay, friends, listen carefully. Now, virus हा सेल मध्ये आलेला आहे. आता त्याच्यावरचं कव्हरिंग काढायचं पाहिजे. So it is the process in which capsid is removed and nucleic acid is released into the cell cytoplasm. Okay, वरचं कोट रिमूव्ह झालं and आता जो न्यूक्लिक ऍसिड आहे, that nucleic acid now is free in the cytoplasm. The nucleic acid of plant virus enters the host cell cytoplasm along with capsid. Okay, capsid सोबतच एंटर होतं, नंतर ते मोकळं होतं. In the cytoplasm, capsid is removed and nucleic acid is free. Okay, it requires assistance of host enzymes to remove the capsid. Here, in this process of uncoating, host helps. Okay, host helps to uncoat the virus particle. Host enzymes. As far as virus whole is concerned, viruses have very few enzymes with them, but most of the enzyme system of host is used by the virus, isn't it? Okay, host cell ची बरीच एन्झाईम सिस्टम हे व्हायरस वापरतं. व्हायरस कडे खूप कमी एन्झाईम्स आहेत, ते एकदम मोजके एन्झाईम्स आहेत, isn't it? आणि तेही होस्टच्या मदतीनेच काम करतात. Okay, next. Now, cellular development. So, in order to produce disease, the virus must replicate and spread to the neighboring cell and then systematically throughout the plant. Okay, so for the development of disease, virus needs to spread inside the cell. The spread of virus into the neighboring cell occurs through the microscopic channels in the cell wall that is called plasmodesmata. So, what are plasmodesmata, friends? Plasmodesmata are the cytoplasmic connections present in between the adjacent cell. एक सेल त्याच्या शेजारी सेल सोबत कम्युनिकेशन मध्ये असते, कॉन्टॅक्ट मध्ये असते. So that communication is maintained, or that contact is maintained through the channel, what we call plasmodesmata. Okay, so plasmodesmata is the channel. So there are cellular structures. Okay, so these are the structures which extends from cell wall. Okay, and connect adjacent plant cell. म्हणजे एक plant cell दुसऱ्या plant सोबत या plasmodesmata ने connect करते. So through this plasmodesmata, virus passes. The spread to the other part of the plant takes place through xylem and phloem. Okay, we know xylem and phloem are the, what you can say, conducting tissues, vascular tissues. So xylem and phloem. Xylem helps in the transport of water, whereas phloem helps in transport of food material. So through this xylem and phloem, the further spread of the virus throughout the plant takes place, occurs there. The cell-to-cell movement of virus requires one or more protein that are called movement protein. Okay, एका सेल मधून दुसऱ्या सेल मध्ये व्हायरसला जाण्यासाठी काही प्रोटीनची आवश्यकता असते, and that proteins are called as movement proteins. Most plant viruses use coat or capsid proteins for transport. Okay, आता बऱ्याच ठिकाणी जे प्रोटीन कोट्स आहेत, कॅप्सिड आहे, त्याचाच वापर त्या ठिकाणी केला जातो. Okay, next. Now come toward the cellular development again. So we'll go deep into the cellular development.
चल कोण कोण मी
Cellular development. Now TMV contains single stranded RNA as genome. Okay, nucleic acid हा single stranded RNA मध्ये आहे. That is called तो स्वतःचा जो RNA आहे ओरिजिनल RNA स्वतःचा त्याला पॉझिटिव्ह RNA म्हटलेला आहे. ओके, जो ओरिजिनल TMV व्हायरस मधली जो काही RNA आहे त्याचा that RNA single strand RNA is called positive strand. That is called positive RNA. Okay, so replication of virion or virus RNA thus involve synthesis of negative strand using positive strand as a template. Okay, एक पॉझिटिव्ह स्ट्रँड आहे त्याच्या बाजूला निगेटिव्ह स्ट्रँड बनतो. ओके, वी नो इन द प्रोसेस ऑफ डीएनए रिप्लिकेशन टू स्ट्रँड्स गेट सेपरेट आउट आणि एका स्ट्रँडवर दुसरा स्ट्रँड बनतो आणि या स्ट्रँडवर दुसरा स्ट्रँड. सो मेसर्स्टाल एक्सपेरिमेंट तुम्ही पाहिलेला आहे आणि त्याच्यामध्ये सांगितलं की डीएनए सेमी कॉन्झर्वेटिव्ह रिप्लिकेशन होतं, म्हणजे एक स्ट्रँड असतो आणि त्याच्या बाजूला दुसरा स्ट्रँड बनतो. ओके, जो पहिला स्ट्रँड आहे दॅट इज कॉल्ड पॉझिटिव्ह स्ट्रँड आणि जो बनणार आहे त्याला निगेटिव्ह म्हणायचं, बिकॉज पॉझिटिव्ह त्याला कॉम्प्लिमेंटरी न्यूक्लियोटाइड अटॅच होत असतात बाजूला. ठीक आहे, सो धिस प्रोसेस रिक्वायर्स so through this process the entire new strand develops and that new strand is nothing but negative strand. Thus replication complete in two steps. Now main part is the replication of RNA. याच्यामध्ये RNA चा replication चा main part आहे. The synthesis of negative strand using RNA. Okay, synthesis of negative strand RNA using a positive strand as a template which forms double stranded intermediate termed as replicative form. ओके, आता बघा एक रिप्लिकेटिव्ह फॉर्म त्या ठिकाणी म्हणतो. आता याच्या पुढच्या स्लाइड मध्ये तुम्हाला अजून डिटेल कळून जाईल. सो आय विल रिपीट दिस. सिंथेसिस ऑफ निगेटिव्ह स्ट्रँड आरएनए म्हणजे नवीन बनणारा आरएनए तो निगेटिव्ह आहे. व्हॉट? युजिंग पॉझिटिव्ह स्ट्रँड आरएनए ॲज अ टेम्पलेट which forms double stranded intermediate termed as replicative form. RF रिप्लिकेटिव्ह फॉर्म आधी बनतो आणि रिप्लिकेटिव्ह फॉर्म नंतर त्याचे मल्टिप्लिकेशन होतात. ते पुढच्या स्लाइड मध्ये तुम्हाला कळून जाईल. अँड सिंथेसिस ऑफ पॉझिटिव्ह स्ट्रँड. आता ठीक आहे, तुमच्याकडे निगेटिव्ह स्ट्रँड आरएनए आहे त्याच्यापासून पॉझिटिव्ह स्ट्रँड म्हणजे जो आरएनए म्हणजे मेन आरएनए आहे तो पण बनला पाहिजे ना. ओके, सो दॅट सिंथेसिस ऑफ पॉझिटिव्ह स्ट्रँड आरएनए युजिंग निगेटिव्ह स्ट्रँड आरएनए ॲज अ टेम्पलेट. सो दॅट using that here virus codes this virus codes RNA dependent RNA polymerase. Okay, so I will repeat this one. Synthesis of positive strand RNA using negative strand RNA as a template. So this is using virus coded RNA dependent RNA polymerase. आता व्हायरसच बनवतो. काय बनवतो? व्हायरस कोडेड RNA हा बनवण्यासाठी म्हणजे दुसरा पॉझिटिव्ह स्ट्रँड बनवण्यासाठी हा व्हायरसच आरएनए पॉलिमरेज बनवतो. म्हणजे हा म्हणजे आरएनएच आरएनए पॉलिमरेज बनवतो. आरएनए कोडेड व्हायरस कोडेड डिपेंडंट आरएनए पॉलिमरेज. सो दिस सेंटेन्स इज इम्पॉर्टंट. ओके, व्हायरस कोडेड आरएनए म्हणजे व्हायरसनच आरएनए बनवलेला आहे आणि त्या कोडच्या आधारे आरएनए पॉलिमरेज बनतो. ओके, नेक्स्ट पॉइंट पुढे बघा. हा, yes, so you will get here. This one is replicative form, friends. RF replicative form. So friends, you see here in this first diagram here, this is the RNA. We call it as positive RNA. पॉझिटिव्ह म्हणजे जो मेन आरएनए आहे कुणाचा? TMV चा, व्हायरसचा. So that is a positive RNA. So this is positive RNA. So on this RNA, so this RNA strand acts as a template, black colored RNA acts as a template, and on this template negative RNA strand is developed. So that RNA is called as negative RNA, and this total structure is called a replicative form of RNA. तर या याच्यापासूनच रिप्लिकेशन होणार आहे पुढे. This is called replicative form of RNA, and from this replicative form here multiple copies of RNA are formed. Okay, हा कोणता RNA आहे? This RNA is what? Negative strand RNA. Okay, negative strand. Now, so from this, here come to the second diagram here, this side. So from this RNA, here blue, green color RNA, what is the color? Okay, so this negative strand RNA again produces positive strand RNA, because the positive stranded RNA is important. Okay, so this needs to be synthesized. Positive strand RNA, black color here, this is black color, positive stranded RNA. याच्या मल्टिपल कॉपीज झाल्या पाहिजेत. या पॉझिटिव्ह स्ट्रँड आरएनएच्या मल्टिपल कॉपीज तयार झाल्या पाहिजेत, so that for the final assembly of virus, here these positive copies of RNA requires, isn't it? Okay, जे काय व्हायरस पार्टिकलची फायनल असेंब्ली असते, त्या असेंब्लीसाठी हे पॉझिटिव्ह कॉपीज गरजेचे असतात. सो दिस पॉझिटिव्ह आरएनए. ओके, म्हणजे काय, अगोदर निगेटिव्ह आरएनए तयार झाला, निगेटिव्ह वर पॉझिटिव्ह तयार झाला. Once this negative RNA is formed, negative strand RNA formed, so now you see here how many negative copies are producing. That producing in many number, multiple number. Okay, multiple negative RNA बनतात. आता जेवढे काही मल्टिपल आरएनए बनलेत त्या प्रत्येकावर पॉझिटिव्ह आरएनए बनतो. ओके, and at the same time, once the positive RNA forms, at the same time here subgenomic RNA produces capsid protein. Okay, we know RNA is directly involved in the process of translation. Okay, RNA is involved in the process of translation. You know the process of protein synthesis. In case of DNA produces RNA and RNA produces protein. DNA produces RNA, this is called transcription, and RNA produces protein, this is called as translation. Okay, so here RNA is there. So from this RNA directly translation occurs, means synthesis of protein occurs. So what are those proteins? Those proteins are capsid proteins. Okay, so these are many in number, हे खूप जास्त असतात. So capsid proteins are the proteins required for what? Say कशासाठी पाहिजे? So these proteins are required for final assembly. Okay, so these proteins are capsid proteins are required for final assembly, packaging of rod shaped virus. Okay, so this is very important, friends. Okay, so this is the process at molecular level, multiplication of TMV. So in your syllabus general multiplication is mentioned. सिलेबस मध्ये फक्त मल्टिप्लिकेशन ऑफ व्हायरसेस असा शब्द प्रयोग केलेला आहे. बट ऍज अ पार्ट ऑफ प्लांट सायन्सेस, ओके, आपण प्लांट सायन्सचे विद्यार्थी असल्यामुळे, बॉटनीचे विद्यार्थी असल्यामुळे आपण प्लांट व्हायरसेसचं मल्टिप्लिकेशन घेतो त्या ठिकाणी. ओके, जनरल व्हायरसचं मल्टिप्लिकेशन वेगळं आहे, म्हणजे वेगळं म्हणजे ते वेगवेगळ्या व्हायरसचं मल्टिप्लिकेशन प्रोसेस वेगवेगळी आहे. टीएमव्हीची वेगळी आहे, बिकॉज टीएमव्ही प्लांट व्हायरस याची प्रोसेस वेगळी आहे. अॅनिमल व्हायरसची कदाचित प्रोसेस वेगळी असेल, बॅक्टेरियोफायची प्रोसेस वेगळी आहे. Isn't it? So basically major theme एक असं minor differences आहेत प्रत्येकामध्ये. So here once your genomic RNA and capsid proteins are formed, so they are in multiple number. जेवढे इकडे RNA बनतात तेवढे त्याच्याकडे कॉपी बनतात. Okay, now the final process is assembly. Assembly, this is also called as maturation or assembly. So after the intracellular synthesis of enough number of capsid proteins and RNA, so assembly occurs, assembly begins. Assembly means packaging. It is highly organized process. Okay, he खूप organized process है. Initially the capsid assembly begins when capsomer associate with 3' end of RNA. Okay, so friends, RNA has 3 ends, 2 ends, 5' and 3'. Okay, त्याला आपण 3' आणि 5' म्हणतो. DNA ला 3' and 5' किंवा 5' 3'. एन टोक असतात त्याचे, ठीक आहे. सो असेंबली बिगिन्स ऍट 3 डॅश एंड, त्याला 3 प्राइम एंड, ओके 3 डॅश म्हणून त्याला, किंवा 3 प्राइम एंड ऑफ आरएनए. आरएनएचा हा लॉंग स्ट्रँड असतो, त्याला 2 टोक असतात, एका टोकाला 3 डॅश एंड म्हणायचं, दुसऱ्या टोकाला 5 डॅश एंड म्हणायचं, किंवा 3 प्राइम एंड आणि 5 प्राइम एंड. ओके, सो दिस इज व्हॉट द टर्म, हे टर्म वापरतो आपण, किंवा त्याला प्राइम म्हणायचं. सो ऍट 3 प्राइम एंड असेंबली बिगिन्स. ओके, सो टीएमव्ही असेंबली ऑल्सो अकर्स इन विट्रो. बघा, लॅबोरेटरी मध्ये पण टीएमव्ही असेंबली करता येते when the capsid proteins and nucleic acid are mixed together. ओके, एका टेस्ट ट्यूब मध्ये तुम्ही कॅप्सिड प्रोटीन आणि हा न्यूक्लिक ऍसिड जर मिक्स केला, ओके, जर एका टेस्ट ट्यूब मध्ये कॅप्सिड प्रोटीन आणि न्यूक्लिक ऍसिड मिक्स केला इन लॅबोरेटरी, at that time you will get final assembled TMV virus. Okay, so we will find final assembled TMV virus particle. So this is the process of assembly. Okay, assembly म्हणजे या व्हायरसचे पूर्ण रूप बनणं. सो व्हायरस तयार होणं पूर्णपणे, हे दिस इज हॅपनिंग, दिस इज ऑकरिंग इन द होस्ट सेल. Okay, assembly occurs in the host cell. Now, the final step, friends. So this is very important again. So likewise, in the cell, multiple copies of TMV are formed. Okay, TMV च्या ह्याच्यामध्ये मल्टिपल कॉपीज, या होस्ट सेल मध्ये TMV च्या एका सेल मध्ये TMV च्या भरपूर कॉपीज तयार झाल्या. मल्टिपल कॉपीज. सो प्लांट cell now, as a result of assembly and multiplication, here the plant cell acquires multiple copies of TMV. लाखो कॉपीज त्या ठिकाणी एका सेल मध्ये बनतात. आता सेल मध्ये जर या कॉपीज बनल्या तर व्हॉट हॅपन, द सेलला स्वतःचं काम करायसाठी जागा राहणार नाही. ओके, सेलला स्वतःच काम करायसाठी काय जागा राहणार नाही. सो द सेल विल फेस व्हॉट यू कॅन से स्केर्सिटी ऑफ स्पेस. जागा कमी पडेल. सेलला स्वतःच जे काही काम करायचं आहे ते काम करता येणार नाही. अल्टिमेटली इट विल लीड्स इनटू द डेव्हलपमेंट ऑफ सम सिम्पटम्स. Okay, so what happens, as a result of formation of more number of copies in the cell, the cell itself cell हे मरते. And how this happens, virus particles comes out by lysing, और त्याला म्हणतो बस्टिंग. ओके, बस्ट करणे, लाइज करणे, सेलला ब्रेक करणे, ब्रेक करून हे व्हायरस पार्टिकल से सेलच्या बाहेर पडतात. अँड धिस लीड्स इनटू द किलिंग ऑफ दॅट होस्ट सेल. ओके, द प्रोसेस ऑफ किलिंग ऑफ होस्ट सेल इज कॉल्ड एज नेक्रोसिस. ओके, होस्ट सेल डेड होणे म्हणजे नेक्रोसिस होणे. तिथं त्या पानावर, त्या लीफवर यू विल गेट सिम्पटम. तो भाग वाळतो, ड्राय होतो. ओके, पानावरचा काही भाग आधी पिवळा पडतो आणि लॅटर तो पिवळा पडल्यानंतर हळूहळू पुन्हा तो ड्राय होतो आणि तिथल्या सेल सगळ्या मरून जातात. सो दिस इज द प्रोसेस. सो व्हायरस पार्टिकल्स, assembled virus particle releases out of the cell by the process of autolysis of host cell. प्लांट सेलच्या बाहेर ह्या सगळ्या व्हायरस पार्टिकल येतात. सो फ्रेंड्स, दिस इज व्हॉट द टोटल प्रोसेस ऑफ मल्टिप्लिकेशन ऑफ टीएमव्ही. ओके, अशा पद्धतीनं टीएमव्ही व्हायरसचं